So we have the pleasure to host Kevin Painter from Politecnico di Torino, and he will be talking to us about extensions of the Keller-Sager framework. So, Kevin, let's start.
Thank you very much for the introduction, and thanks very much to the organisers for the invitation. So we're going to be talking today about something that I've really been working on for a lot of time, and that's kind of models of hematoxins, basically, and in particular two extensions to the kind of the standard Keller-Siegel framework, which I think will be probably familiar to quite a lot of those people in the audience, but I will obviously have a few lines of introduction. So this kind of comes into the kind of basic sort of observation that pretty well all organisms detect and respond to external chemicals. So from the kind of single cells to large organisms, and this has been kind of studied intensely as a problem, as a kind of problem for many decades, indeed even sort of centuries. And it's well known that certain animals have incredibly sensitive sense of smell. This sort of picture here is showing a gun dog, essentially, and basically it's there's a kind of a dead pheasant that has been dragged across a field. The dog has been released somewhere up there. There's a kind of light on it, so recording its movement. And we're kind of able to kind of see this kind of dog kind of search through, even despite the kind of the kind of limited visibility, follow the trail of this pheasant, and obviously eventually sort of finds it. And that's obviously a kind of an incredibly important ability in order to locate food sources. And this is an ability that certainly dogs are very well known for their kind of extraordinary sense of smell, but of course it is actually also something that we are able to do as humans as well. Because actually if you kind of take a kind of a willing PhD student and lay down a trail of chocolate in the field, then maybe not quite at the same speed as the dog, but they are also able to kind of follow through and find the follow through that kind of line laid out in the field. So this kind of sense of smell is something that is kind of pretty well ubiquitous across the animal world. Now, this is and another kind of another nice kind of study on kind of humans also can be kind of shown in sort of young babies, basically. So these kind of very kind of early crawling babies, about sort of six to 9 months, basically, and they have actually incredibly strong olfaction. At that stage, the sight is a little bit less developed, basically, and they will respond to the odours released by the mother, basically. And they've done studies, basically, where kind of dress pads have been kind of left in the room, either unused or in used format, and the unused one, the baby just simply pretty well ignores it. They kind of just move around in a kind of a random walk type sort of direction. But if you actually use used breast pads, which obviously have the scent of the mother on them, then the baby is able to pretty well kind of take a kind of a direct line towards those breast pads. So this is a very innate ability that we have as humans to detect and respond through movement to chemical signals. Now, this induces a lot of behaviours, and probably one of the kind of the areas that has been most popularly looked at, certainly within the mathematical biology community, is the ability to induce aggregation. And various organisms or cell populations have shown the ability to self-organise into groups through modulating their chemical environment. Dictyostelium is the classic paradigm of such an organism, these kind of single-celled organisms that transform themselves into a multicellular organism, and they do that through aggregating in response to self-produced chemical PAM. But it's also seen in large organisms as well. These are spiny lobsters that aggregate together into groups on the bottom of the ocean floor through producing and aggregating pheromone. So, and that's the sort of phenomena that I'm going to be looking at in the first part of today's talk. And the textbook model for this is that was produced by, that was kind of developed by Keller and Siegel in the early 1970s. And I think, again, a lot of the audience will be familiar with this. It's, there's various sort of extensions of this, but in its kind of essential form, it has a population, which could be either cells or organisms. They responding to some sort of attractant, and this term here, this kind of directed movement term here, is describing a chemotaxis response. So the population responds and moves according to the gradients of the chemoattractant. And as I say, this was a model introduced by Keller and Siegel in the early 1970s. We typically call alpha the chemotactic sensitivity, and that would be positive for chemoattraction. And it's been a model that's been applied in a lot of different areas, both from its original roots in microbiology, but also in areas such as developmental biology, ecology, and even areas such as sociology and economics. And I just had there's kind of I wrote a relatively recent review on this applications of this model in diverse areas a couple of years ago. So that's the textbook model, and in particular, this model has been extensively studied for its ability to show aggregation, autoaggregation. And again, this is a kind of a particularly well-known formulation. You kind of call it, in a sense, a minimal autoaggregation mechanism, basically, where you have a population that is moving up the gradients of the chemical and is also producing the chemical. And this positive feedback is the key to aggregation in this mechanism, that the population is both producing and responding to the attractant. And a classic Turing type linear stability analysis will show that this model is capable of inducing autoaggregation, so that is aggregation from an effectively uniform initial distribution, according to the kind of the parameters of the system. And I think, again, it's probably a lot of you know that there's very big literature on this sort of model, both regards to the applications of it, but also the very interesting mathematical dynamics of this. So that's kind of our basis, and essentially, as I say, you know, what this kind of what this autoaggregation does, you can kind of write down the kind of parameter space for this model, and if the chemotactic sensitivity represented in this kind of alpha parameter here, and the kind of the population density is sufficiently strong, then you are capable of generating autoaggregation, so a dispersed population can aggregate together. And essentially then you can take parameters in that system, start your population in a relatively uniform distribution, then through this positive feedback of climbing up gradients and producing the attractant, you get the organization of the population into a number of clusters. And as I say, that kind of gives the kind of the root of autoaggregation in this sort of palisade model. So that's kind of the basic introduction. And what I want to kind of use this talk for, well, it depends a little bit on time, so whether or not I get to part 2 will depend on how long I can talk in the first part of this talk. I'm going to talk about 2 extensions that I've been working on regarding this framework. And the first is the impact of neotaxis and flow on chemotactic aggregation. And then the second one, if I have time, is the incorporation of phenotypic heterogeneity. So those are the kind of the 2 extensions that I'm going to talk about. And as I say, I'll start with this first one, and this is from recently published work from last year. So what's the motivation for this first study? Well, the motivation really kind of comes from aggregation phenomena such as this. So, you know, this is an aggregating ball of krill. These are these spiny lobsters that I was talking about earlier. And this is a kind of a population of a certain type of fish. I'm not quite sure what they are. But these are all examples of aggregations that are occurring in fluid environments. And there's various reasons that organisms want to do that. But one of the kind of probably the most kind of prevalent of reasons is for what's called broadcast spawning. So a very common method of reproduction in marine organisms is for the population to group together and simultaneously release the sperm and egg cells. And in this kind of group form, because the sperm and egg cells are being directly released into the water, doing this in a kind of a large group should hopefully increase the possibility of fertilization. So many organisms, as I say, need to aggregate in a fluid environment. And this also goes down to kind of the microscopic scale as well, even down to very, very small organisms, this sort of aggregation type phenomenon in a fluid. That then, of course, raises the question that fluid environments are, of course, highly turbulent and volatile. In a kind of simulations of kind of ocean dynamics, you have highly complicated fluid dynamics going on, prevailing currents, vortices, etc., etc. So you've clearly got a highly kind of dynamic fluid environment, and it's quite conceivable to think that that could have a range of both positives and negatives on the ability to sort of form and maintain a group. Benefits from being in a kind of a fluid environment is that the kind of flows may help to coalesce individuals or help to transport the signal, but of course detrimental ones could mean that it could kind of dilute the signal or indeed even kind of split clusters or break them apart. So basically, as I say, we kind of like to understand, when you've got either positives and negatives emerging from this kind of bowl of fluid on the ability to form and maintain a cluster. So that's kind of the effect of flow. But of course, animals that have that live in fluid environments have also developed ways to counteract that flow. And in particular, kind of a very well-known mechanism is what's called rheotaxis. And I'll kind of show a video here of rheotaxis in a kind of a moment. But essentially, rheotaxis is the orientation of movement according to currents, basically. And to demonstrate this, I'm going to show that this is from a kind of an experiment on the same species, so 3-spine stickleback. They're a small fish of about maybe 2-3 centimeters in length. And the population in the right has been a population that were born in a stream environment. So they were kind of born in an environment where there's a pretty consistent current in their environment. The population in the left are from a population born from a lake, so where the currents are probably slightly less strong on the whole. And they basically put these two populations into a circular tank here. There's a little motor here that generates a flow in an anti-clockwise direction. And then when you kind of see the behaviour, you see that the stream stickleback are orienting themselves according to the flow and effectively therefore maintaining positions. That lake population, on the other hand, even though they do show a certain amount of orientation with response to the flow, they've clearly got they're less able at doing it. But essentially, anyway, what this kind of shows is that organisms that are used to living in a fluid environment have developed countering ways to counter the effects of flow. Rheotaxis, as I say, is found in a lot of different organisms. It's found in the same, pretty well all fish species show the ability to perform rheotaxis. It's also shown in things like kind of turtles, in jellyfish, various other marine organisms. Rheotaxis is also something that you see in single cells as well. And indeed, a kind of famous example is in terms of human sperm. And again, if you kind of take human sperm cells and subject them to a flow, you actually see them orienting themselves according to that flow field. So it's also seen in single cells. And I believe it's also been observed in epithelium cells. It's also been observed even in certain cancerous cells as well. So it's a very ubiquitous phenomenon of both single cell and cell. So, in the first part of the talk, what I want to talk about is how do these kind of two factors of flow and rheotax impact on the ability of a population to both form and then maintain a clustered state. So that's the question I want to address in the first part. So to do that, we take a fairly straightforward extension of the Kenner-Siegel model. So basically, we kind of just take that kind of earlier model earlier. I've kind of taken a slight adaptation. I've kind of used this kind of volume filling effect to kind of, prevent sort of severe aggregations in sort of forming this system as well. So essentially, it's just an extension of that minimal model earlier. But we've introduced a flow and we've introduced a rheotactic response, which of course is also oriented according to that flow ratio. So that's a very sort of straightforward extension to the model that we've introduced earlier. And I'm not really going to actually specify a specific equation for the flow field as well. Because there's been various studies where there's also this is then coupled to kind of an Navier-Stokes type equation for the fluid flow dynamics here. But we're just going to prescribe that flow field in these sort of initial studies. But essentially, as I say, what we've got here then is W is my flow field, alpha is still my measure of the chemotaxis mechanism, but the critical kind of new parameter in this system is the rheotactic response, and you can kind of consider the value of that according to in certain ways. Setting phi to 0 would just correspond to negligible rheotaxis, so we have no rheotaxis response there. The population, the attractant would simply be affected by the flow. If I set those phi's equal to 1 here, then of course that rheotactic response for the population will directly counteract the flow. So, you would have then kind of a flow in the attractant, but not in the population. And then other regions can be considered as either undercompensating or overcompensating rheotaxis according to the sort of reference for values. So that's the model that we're going to be looking at. And I really now kind of just want to kind of go through a number of observations then that we noted when kind of studying this. And the first one is when you add rheotaxis into this system, then it has the effect of slowing or preventing autoaggregation that's induced by chemotaxis. So this can be done through a linear stability type analysis, basically. So you can do a standard linear stability analysis. I won't go through the details, but essentially, if you kind of take a sort of parameter set down here, where you've got a relatively small amount of flow and rheotaxis in the system, then you'll generate patterns within a certain characteristic time frame. Install a non-dimensional model where for the 30 time you've seen the forming of the aggregates here. But when you kind of add extra flow or extra rheotaxis into the system, the aggregates take much longer to form. Or you can even be in a kind of a case where, as you increase the amount of rheotaxis or flow into the system, it pulls you out of the autoaggregation space, and you're no longer able to autoaggregate. So a kind of linear stability analysis indicates then that they're kind of adding rheotaxis into this framework can slow or even prevent cluster growth. And the intuitive reasoning for why this is happening is that because when you put in flow into the system, the signal is going to be transported in the flow. The rheotaxis brings the population away from that, in the opposite direction to that current, so away from those aggregations. So it will kind of slow down that flow. Okay, so our kind of first observation was adding rheotaxis slowed down the rate at which clusters can form. The second observation was looking then at post-aggregation dynamics. So while rheotaxis slowed down the initial formation of clusters, we found, though, that post-aggregation, adding rheotaxis, accelerated the subsequent unification of clusters. So I'll explain what I kind of mean by this, with this sort of dynamics. A well-known phenomena in chemotaxis sort of models, Keller-Siegel chemotaxis models, is this sort of coarsening type behavior you emerge. That in the initial stage, you develop a number of aggregates that are separated by some characteristic wavelength. But then, because these clusters are still producing attractant, the clusters will attract each other, and over time, they will slowly coalesce into fewer and fewer clusters. So this kind of coarsening type sort of phenomena that is associated, that you find associated to chemotaxis type models. But the time scale of that could be very, very large, basically. And in fact, in this sort of case here, where there's no rheotaxis in the system, you see that you kind of, in the initial stage, you form, well, say about sort of 7 or 8 clusters here, but then, you know, right through at the end of the simulation, you've still got the same number of clusters. That time scale over which that they unify can be much longer, you know, the actual, can be very, very long with respect to the time dynamics of initial aggregate formation. When you add in rheotaxis, though, you get a much more accelerated unification time, basically. So as we move here from kind of no rheotaxis to compensating rheotaxis, you see a very quick unification of all of the clusters into a single aggregate. And this would be advantageous for a population that really wants to form a very large aggregate, collecting together as many as possible. So rheotaxis has this effect of accelerating unification time. And the reason for that is basically because rheotaxis will act, the effect of rheotaxis is different according to the cluster size. So to test this, I've started with a population with 3 different sized clusters, so one located here, one located here, one located here, and then each of those has got a different sort of mass, so 4, 2, and 3, etc. And you can see that when kind of, even though they're all performing the same level of rheotaxis, though they've got different abilities to maintain themselves against the flow. This smaller mass one is still transported quickly by the flow. The larger mass is more able to hold its position. And the consequence of this is then that the smaller masses get swept into the larger masses, and over time cluster into a single large mass that is able to hold its position against the flow. So those are our kind of first 2 observations. Both of those within the context of a very simple, uniform flow type environment that allow us to kind of do this in a kind of slightly more analytical way. But what about when we kind of move into the kind of the real world relevant scenario of a variable flow? So to look at variable flows, in the first instance, I'm going to look at a very controlled variable flow where we interrupt kind of a uniform flow field with a vortex type structure. So t less than 100 and then t greater than 300, you just have my flow as being uniform, pointing from left to right. And then for the intermediate time, we're going to put a kind of a vortex type structure in the middle of this flow field and see how that impacts on the dynamic population. Okay. So this is our kind of next observation, and that was that a variable flow will seed the clustering even within what we call these kind of low density or weak chemotaxis regimes. Okay, so what I mean by this is as follows. If we go back to that kind of auto aggregation parameter space, we're going to select a parameter which is outside the auto aggregation region. So if the population is dispersed, for these parameters, the model will not be able to kind of aggregate aggregate into clusters through this chemotaxis mechanism. And you can see that then in my population, that while we're in this kind of uniform flow field, the population is remaining dispersed. But when you add in then the vortex has the effect of coalescing individuals in a kind of a low flow regime. And once it's done that kind of can effectively trigger this auto-aggregation through chemotaxis and form into a kind of a large cluster. And then whenever you take away that vortex and return to the uniform, that large cluster can maintain itself and then, effectively maintains itself over time. So essentially, what's kind of happening there is that this kind of variable flow will have the effect of amplifying the parameter space where patterning can occur. So what's the reason for this? Well, to investigate this, we did a kind of a numerical bifurcation analysis here. We idealized this into a one-dimensional case, and the root cause appears to be down to the bistability mechanism that you have in the chemotaxis auto-aggregation system. So in a case of a uniform flow, and where we've got no chemotaxis in the system, so I should say actually these results at the moment are without chemotaxis, so we're not having any chemotaxis effect, we're just looking at the effective flow. In this sort of case, the bifurcation sort of picture for the kind of SQL model can be kind of condensed into the following sort of picture. You've got a kind of a lower branch steady state, which corresponds to kind of a uniformly distributed population. And then you've got these kind of upper branch states that correspond to a pattern solution. And basically, this kind of lower branch state remains stable up to some alpha star, and this is the value that you get from your linear stability analysis. And when you increase alpha star above alpha star, you kind of go up into this kind of stable upper branch where you've got the aggregated solution. But this branch extends both sides of alpha star. So essentially, you have this kind of region of bistability where an aggregated solution can actually remain stable down to some lower threshold at alpha double star. You've got this bistability region where both the uniform solution and the aggregated solution are stable for the same chemotactic sensitivity. So that's a kind of a uniform flow scenario. But when you kind of go into a variable flow, where you kind of maybe have kind of a region of kind of slower flow, such as here, then what that effectively does is it kind of reduces the point at which you get in, where you get movement up, where you kind of lose the stability in this kind of lower branch and move onto the aggregated solution. So that kind of triggers clustering in this kind of weak chemotaxis regime. You get into the cluster form, and then even if you then remove that flow, you remain stable. But, you remain in this kind of stable regime for the clustered solution. So you can understand that a little bit through kind of the bistability in the chemotaxis mechanism. Essentially, what this is kind of saying is that as when you kind of add in a kind of a variable flow region, it has the effect of it amplifying the parameter space in which stable aggregates can form. So variable flow, can actually help to encourage pattern formation by providing sort of seed points from which clusters can then form. So how does rheotaxis enter this? Well, if you add then rheotaxis, it negates this positive benefit of flow. And you can kind of see that in the following case. So this is my no rheotaxis sort of scenario where you go from a uniform to kind of a vortex solution that kind of encourages a cluster to sort of form. If you do exactly the same sort of setup, adding rheotaxis, then we no longer get my clusters formed. And again, what's kind of happening in this sort of case, again, you can kind of study this through a kind of numerical bifurcation analysis. When you're adding rheotaxis. Rheotaxis is countering the effect of flow, so that effectively has this kind of, counters that earlier benefit, that kind of threshold at which you get that kind of the instability of this kind of lower part, which is kind of raised back up towards the original instability. So rheotaxis then prevents this sort of beneficial effect of flow in transsucceeding clusters. Okay. So those were our kind of first observations when we looked at variable flows. In that variable flow can kind of encourage pattern formation by single clusters, but then when you add rheotaxis to that, that negates that and negates that sort of benefit. So that's a kind of a very controlled flow. But what if we now kind of look to a much more realistic environment? And the next observation was probably quite an intuitive one, was that if you have a very strong turbulent flow, then that can act to fragment and disperse routes. So we put a more realistic flow environment in. This is a population that's already in an aggregated form, basically, and you put the flow in, but the flow, there's no rheotaxis in this system, and the aggregate gets really kind of stretched and deformed by the flow environment, and kind of gets dispersed, and some of these aggregates end up just simply dispersing outwards. So, when you put in a kind of a strong realistic flow, and no rheotaxis into the system, that flow can have a dramatic effect on the ability of the population to maintain a clustered form. So, strong complex flows can fragment and disperse clusters. But if we then add rheotaxis back into that, then it can provide a stabilizing influence that allows the cluster to maintain itself in the face of turbulence. So that same flow field, the same sort of chemotactic sort of parameters, but looking at the no rheotaxis against the rheotaxis sort of case, rheotaxis counteract effects of flow, and the population maintains nicely clustered and within its kind of original location. Okay, so cluster integrity can then be rescued through rheotaxis. With those observations, basically, what this has kind of led to is the realization that flow and rheotaxis can have both positives and negatives in terms of formation and maintenance of clusters. So flow can, for example, the advantage is it can generate these cluster speeds that kind of encourage pattern formation, even in scenarios between this kind of weak chemotaxis, which normally wouldn't cluster. But then on a kind of negative side, it can kind of fragment and disperse clusters. Rheotaxis has positives in that it can unify clusters, it can sustain clusters, but then it can also have a disadvantage in that it inhibits cluster formation. If we look at where these are happening, the pros of flow are happening in the pre-clustering stage and the cons in the post-clustering phase. For rheotaxis, that's reversed. The pros are in the post-clustering phase and the cons are in the pre-clustering phase. So as our kind of hypothesis, we then suggested that perhaps the optimal response to form and maintain a cluster would be to perform rheotaxis according to your population density. So we extended our model to this form here to introduce density-dependent rheotaxis. And essentially what this does is once the population density passes a critical density, rheotaxis becomes switched on. So it should be switched on in that kind of pre-clustering phase, but it should be switched on in the post-clustering phase. And when we do that, well, just to kind of look, this is our kind of constant compensating rheotaxis, so no density dependence here. You simply don't get any clustering happening. You form very shallow aggregates that they get dispersed and fragmented very quickly. So there's no density dependent rheotaxis there. with negligible rheotaxis, so again, no density-dependent chemotaxis, you can get clusters forming, but they're not very stable. They get kind of disrupted by the flow, they can disappear and then reform elsewhere, basically. But when you have this density-dependent rheotaxis, it does allow you to form clusters and then maintain them against the flow. So it does seem to suggest that this density-dependent rheotaxis could indeed, optimize these pluses and minuses from the flow. Okay, so actually, sorry, can I just ask, because we started a little bit late, how much time do I have left?
I would say maximum 15 minutes.
Okay, that's fine then. Yeah, so
I think it was it was supposed to be 45 minutes, so I propose to give this duration.
Sure, absolutely, yeah. I should only need another 5 minutes or so. 5 minutes. Okay, so that was the first part. The second part of the talk, though, I wanted to kind of work. This is recent work by Tommaso, who is in the audience. And I always like to talk about stuff where there's somebody in the audience and I'm not in the audience, because then they get to deal with any difficult questions from it. So sorry, Tommaso, for giving me that. The second part is then to where a recent study where we've been looking at the incorporation of phenotypic heterogeneity into models of phenotypy. So I'm not going to talk too much about this, but the motivation for this, well, the motivation framework for this kind of goes back right to some of those kind of very classic early experiments in showing the kind of macroscopic dynamics in chemotactic signals, this kind of phenomena of traveling bands of bacteria or spreading rings of bacteria, which you see when you put a inoculated population within a certain nutrient environment, where you see these kind of bands of bacteria that spread through the environment, and as they kind of move through the environment, they consume the nutrient, and that kind of allows these kind of bands to kind of form and then maintain themselves as they spread through the system. And these kind of observations of Julius Agler in the 1960s were one of the kind of key inspirations of Keller and Siegel, and in one of their kind of early, classics of early studies in the 1970s, they investigated exactly that problem. They kind of set up a chemotaxis sort of model to look at these travelling bands and studied the travelling wave, travelling waves in these sort of systems. Now, of course, that kind of early model of Keller and Siegel, though, has a population which is effectively homogeneous. So this population, each individual in that population has the same chemotaxic sensitivity, has the same sort of migratory ability, etc., etc. One of the things that's kind of really been recognised in the last couple of decades is that populations often show a very broad phenotypic heterogeneity. And as an example, if you look at E. coli in certain heme structures, you see significant heterogeneity in their chemotactic sensitivity. You set up a kind of a maze sort of structure here, where there's kind of a nutrient gradient through this sort of maze sort of structure, and then you look at the chemotactic velocity of cells at different points within this sort of system. You see that there's a kind of a varying chemotactic capability, in that the most chemotactic cells are those that have most infiltrated the maze to the deepest sort of point. So there's a strong phenotypic heterogeneity in that chemotactic population. So it's recognised that cells have, effectively the same general population. They can exist in a range of chemotactic. They can have a range of kind of chemotactic sensitivity for values. This phenotypic heterogeneity in their chemotactic sensitivity. But it's also known that if you kind of spend more time doing chemotaxis, then you've got less energy to do other sort of things. And in particular, there appears to be a kind of a negative relationship between whether or not you're doing chemotaxis or whether or not you're growing as a population. You do more chemotaxis, then you're proliferating. And this has also been kind of recognized in the recent study, where you kind of look at the expression levels of the of genes controlling the chemotactic ability, and then negative correlated with the growth rate of the population. So it suggests that, say there's this phenotypic heterogeneity, but where you are in this sort of phenotypic heterogeneity, you will have trade-offs in other parts of in other types of behaviours. So, in order to kind of look at the effect of these sort of things on the dynamics of populations, we've set up an extended kind of SIGL model where we extend to include an additional phenotype space variable Y. So these are kind of similar to models of phenotypic heterogeneity that have been developed by various others, again, people in the audience. But essentially now we have my bacteria population, which is now a function of X and Y, where Y is my phenotype sort of variable. And we've got population responding to some substrate, and basically the chemotactic sensitivity can vary with the phenotypic state of the population. We've got a term here to describe the form of phenotypic switching, which can depend on the substrate. We've got proliferation rate, which again can depend on the phenotype, and we've also got some sort of nutrient attractive assumptions. Okay, so that's the kind of the model that we're looking at. And we decided to look at what we call a chemotaxis proliferation trade-off with escape setup, where I can explain quickly, where basically there's a trade-off between proliferation and chemotaxis. The strongly chemotactic cells are less able at proliferating and vice versa, so that we're kind of choosing that. The proliferation increases with the amount of nutrient. The rate of consumption of the nutrient increases with the amount of the proliferation, and the nutrient level can potentially control this switching of phenotypes. So at low nutrients, the cells can potentially become more chemotactic. At high nutrient levels, they can become more proliferative. And they do that according to the size of this parameter phi, and that can be thought of allowing cells to evade a poor environment by becoming more motile. Okay, so when we look at that, well, the dynamics, when you just have purely random switching, so there's no kind of escaping the environment, you get kind of a classic travelling pulse type sort of behaviour, where you get consumed nutrient environment, travelling pulse type cell density. But this pulse has kind of variability in the phenotypes according to the position of the pulse, where, I should just say, you can do your travelling wave analysis on the system and get wave speed predictions, etc. But basically, as I say, this pulse has a sort of a phenotypic structuring where you get the most chemotactic cells appearing at the front. So they're getting to the front and then they're degrading that nutrient environment, and that's kind of driving the pulse forward. But as you add indirectly switching into this system, then you get a reversal of the situation. You get a more concentrated peak as the rear cells take for nutrient regimes, and effectively you now find the highly chemotactic cells shifted to the rear. So when according to the response to the environment, the positioning in the of the most chemotactic cells of the pulse will change. And then this is somewhat reminiscent of other sort of kind of leader-follower types of systems, where the positioning of the steering cells in the group could change from being at the back of the group to the front of the group according to how they're kind of responding to the environment. What we're finding here is that kind of environmental response will dictate the positioning of the chemotactic cells. Okay, so to summarize then, what I kind of wanted to do is talk about these two sort of general extensions. Rheotaxis and flow, we kind of found that these two phenomena of rheotaxis and flow have opposing effects on the ability to form and maintain a cluster. And our hypothesis is then that if you do density-dependent rheotaxis, that may provide a mechanism to optimize the benefits. In the phenotypic-dependent chemotaxis, we've shown what I've kind of tried to show there was the response to the environment can alter the positioning of the highly chemotaxic cells in invasion rates. And we've got various directions that we want to kind of move this forward in. One is to integrate these two sort of ideas. So in terms of that earlier model that included both rheotaxis and chemotaxis, well, actually, because we kind of assume that they can do both of these at the same time, but it may be the case actually that you have to do one or the other. You either do your chemotaxis or your rheotaxis. So if you have to switch between the two, what's going to be the optimal for forming a group? We also want to kind of look at that kind of phenotypic switching thing and kind of extend it to kind of pattern formation type scenarios as well. And we also want to kind of extend this to a much more general group. To that point then anyway, I'd like to sort of kind of conclude this and say thank you very much for your time.
You can raise your hand in the chat or just tell me. So, okay, maybe I'll start. I have just a question regarding your first pictures of swarming. The swarming of the fishes to reproduce, is it only following some local turbulent flow or are they creating their flow? Because I could see like a trapping effect that could be really interesting for that situation, no?
No,
you create a turbulence, you create a vortex so that you're sure that the eggs and the sperm are located at the same place.
I think that's a really interesting question, actually. As I say, obviously, the moment we make quite a slightly crude simplification that the kind of the movement dynamics of the population have no effect on that local flow environment. I think in the case of fish are obviously a lot more sophisticated, and I can imagine that these fish that you see here, they're kind of big, they're big individuals. They could, through their kind of specifically directed movements of their tail, they could, really be having a kind of a strong influence on that local flow. So, I think, that could indeed actually help to, they could be kind of doing that in a way to really kind of together. It's amazing.
Because it's already a hunting tactic for some predators. I know that orcs are doing that sometimes.
Yeah, absolutely.
They trap fish in the vortex.
Yeah, and it is also even possible so that it's also believed that possibly this is in this kind of krill ball that you're developing here. The krill are probably kind of creating very little local modulations in the kind of the flow environment. I think they collect together already when the kind of the ocean flow is in kind of very still. So they can actually then potentially influence that kind of flow environment. So so the moment we haven't kind of gone into that sort of detail, I think that could be very important. And particularly, I can see it. I can imagine kind of the context of these fish actually kind of very very kind of structured movements could be very crucial to kind of cluster those eggs and sperms together.
Okay, are there any other questions?
We have one question in the room, so we go on to the room question. Sure. Yes, hello. Thank you for your talk. So if you take Keller-Siegel, the classical Keller-Siegel don't add a flow field, one can prove blow up, right? And I think that's clustering, right? That's a way to interpret clustering. So now we add, so I'm at the first part of your talk, right? So now we add this flow field, these parameters. So can we try showing these phenomena you have by also showing blow up there? And because you illustrated it with numerical examples, right?
Yeah, exactly. So I mean, actually, of course, if I take away the kind of flowiness model here, I won't actually get kind of actual blow up because of this volume introduced there, basically. So if you take out, if you set K to 0, you can get blow up, basically. You know, but, I kind of introduced it. This kind of helps the kind of numerics if I keep my population nicely regular. I think it's an interesting question actually about the extent to which when you kind of add in neotaxis, whether or not sorry, maybe I should backtrack actually a moment. I think it would be the case basically if you kind of set your phi to 0, then you should just get a blow up in this sort of model here, you know, as previously. I think that should be the case. When you set phi to be non-zero, I don't know whether or not you'll get blow up or not actually, because at this point the neotaxis is doing something. It's kind of taking, it's going to be taking the population away from the point where the attractant is, you know, so the attractant is being transported and the population is not being transported by the fluid basically. There's going to be a bit of a separation between the two. So I'm not sure how that's going to affect the kind of classical results. I think it's an interesting question, but I haven't looked at it at the moment.
Thank you.
So there will be like quite a decorrelation between the actual attractant and the target that the population is aiming.
Yeah.
Okay, we have a second. Yeah. Yeah, we have a second question.
Okay. Thanks for the nice talk. I would like to know in the reactor, in the reotaxy models. Yes. Is it possible to modify the current of the fluid so as to direct the organism to where we want?
Yeah, so you could, I mean, if I understood it correctly, you're kind of suggesting adding another equation here to describe the fluid dynamics itself. Is that so? And having that depend on the population.
So, with a Stokes model, will be a possibility.
Yeah, I mean, it's something. In fact, actually, people have already been studying kind of fluid dynamic model. We haven't done that in this one because we wanted to kind of concentrate on the simplest possible formulation and just therefore describe fluid dynamics. But yeah, I mean, I think there are very interesting, relevant questions to be looked at when you actually also add in this funding to an equation for W.
Do we have time for an extra question to
It depends how far you want to disrespect the schedule because we're already at the time.
You're the boss, you decide.
Okay, so let's take another question.
Okay.
I'd rather have 10 minutes, 10 minutes late and interact with each other.
Okay, yeah, it's just a very quick question. Thank you very much for the nice talk. I just wanted to know if you have experimental data to confront the reotaxis model and, for instance, to discriminate with if density-dependent reotaxis is more.
Yeah,
you know.
That's a pure hypothesis at the moment, actually. Yeah, I would love to be able to kind of actually have a positive sort of response to that. The problem is it's actually kind of studying these phenomena is extremely difficult to do experimentally in order to kind of really do this. You know, in terms of finding the right sort of system to do this. You can study, you know, they do a lot of the studies on neotaxis in terms of fish, but fish are not probably really using this kind of chemotaxis mechanism as a way to aggregate. They're actually using other sort of mechanisms instead. So, you know, the ideal system would be more if you could find something at kind of a, you know, they're working kind of studying this a little bit quite a long time ago now, since the 70s. You know, unfortunately, those kind of views disappeared at the moment, so I haven't kind of hopefully I can stimulate somebody else to kind of look into this.
Okay, thank you.